we'll take the next talk on glaucoma genetics by Dr. RAJUL PARIKH. So I'm sure we are, all our esteemed panel chairpersons are there. There are enough time to discuss each and every topic.
So I would thank AIOC for the invitation. So I'll be talking about, can we start the?
Yeah, RAJUL, yeah.
Yeah. So I'll be talking about glaucoma and genetics. So I have tried to divide my talk into three parts. So in first part, I'll cover the genetic association. In second part, the role of genetic screening, whether it would be really useful in future. And third, gene therapy. Now, if you look, this is theoretically speaking, the genomic analysis would be the best way to predict that someone genetically is predisposed. Of course, this is the future. I'm not talking that this is the reality at this point in time. Now, when we see the disease, there are two ways. One is the single gene disease and other is complex disorder. That means multiple gene involved. Unfortunately, glaucoma comes in the second category. It's not a single gene disorder. If there's a single gene disorder, then the gene therapy would have been much easier to identify and start. If you look the literature, there are lots and lots of gene loci has been identified, including the chromosome. I'm not going to go in detail because there are multiple. Otherwise, it would just take 10 minutes only to talk about the association. And that is not the part I want to talk about. So basically, we have autosomal dominant inheritance. We have autosomal recessive inheritance. I mean, just remember, we have learned all these things in MBBS. Now, if you look the POAG genes, and I just try to cover a little bit, we have myosin, we have optinurine, TBK1. Now, the mutation, the myosin are associated with 3 to 4% only in POAG with increased intraocular pressure. And if you look optinurine and TBK1, they are associated with only 1 to 3% of open angle glaucoma with normal, that means normal tension glaucoma. Only 4%, not less than that. And so genetic testing, if you do in general for all these, and there are kits available in abroad, I'm sure it will be available in India also. It's not useful because it identifies only 3% of the population with glaucoma. However, if the research has found that the myosin, there are certain variants are associated with very aggressive glaucoma which responds to early surgery. So the research is trying to identify whether which group and whether we can do the testing to identify this group. The research is going on, and this is at this point in time only at the research level, not as a clinical practice level. If you look exfoliation, there are various LOX1 gene, the variants have been identified. I'm not going to go again talk about it. There are protective rare variants also have been identified. If you look, this is from the multi-center study. One of the center was Shankar Netralaya. They identified 3 loci susceptible for the primary angle closure glaucoma. So we have lots and lots of study going on to identify the loci. And the future is interesting. But at this point in time, my question, this is more relevant in day-to-day practice, whether we should use this genetic, what we know in research in clinical practice. So we have to understand that dominant inheritance has a 50% offspring, chances of having the disease going to the offspring, whereas recessive has 1 in 4. So this is the basic thing, So when you do a pedigree charting, and this is very important, then you come to know whether it's autosomal dominant or recessive pattern. Now, for the early onset glaucoma, that is juvenile open angle glaucoma, congenital glaucoma, the association is approximately 20% for the various genes like myosin, PTEX2, FOXC1, so 20%. Now, I will just come to next for American Academy because they come with the guideline that how to use this. So the American Academy guideline says that only if it's going to impact, and this is a basic thing, that only if it's going to impact the clinical practice, then we should use that test. So genetic testing also come in the same pattern. So if the patient with the parents with juvenile open angle glaucoma, congenital glaucoma, it may be useful. POAG, the late onset glaucoma at this point in time is definitely not useful. So, and I just try to give some of the example I borrowed from Ronnie. So this was the one patient with the atypical axon-filled rigor, and I had sent it to Ronnie because they have a much better setup than my private practice. So we wanted to differentiate between axon-filled rigor, whether it's mutation or primary congenital glaucoma. And why it makes difference? Because if the patient has a axon-filled rigor mutation, then chances of going into the offspring is nearly 50%, as opposed to primary congenital glaucoma is little lesser. The same thing, this was one of his patient, and he had given me the slide, that the patient father was juvenile open-angle glaucoma, 3 kids, they did the mutation study, and that one kid had mutation for the juvenile open-angle glaucoma. So that kid should be examined more thoroughly, and other 2 kids can be, we need not examine very aggressively. So this is how you can use, but again, the bottom line is that at this point in time, the genetic screening is basically should be used more for the research purpose rather than day-to-day clinic purpose for the screening. And then I will come to my last part, gene therapy, this is something going to be interesting, because we know medication, we know surgery, everything has limitation, and we also know that there are lots of the mutations happening. So if we can take care of the mutation, the problem, then we can rectify the glaucoma to a certain extent in that particular patient. So this is going to be interesting. So what is gene therapy? So by definition, the medical approach that treats or prevents the disease by correcting the underlying genetic problem. Now there are 2 methods. One is gene transfer and other is genomic editing. So I'll try to cover a little bit of these 2 aspects. So gene transfer, which is going on for some long time. So basically it introduced the new genetic material by various vector, and usually there is various viral vectors are used. Now recently the nanoparticle, the nanotechnology also has come into play to use as a vector. And the third is we can introduce the direct full DNA. So it alters the gene, causes the necessary protein to be faulty or missing. Gene transfer therapy does edits that, and it corrects the gene problem and the protein malfunction. So this is one example. This is not from the glaucoma, but basically you have a viral vector and you add that new gene which you want to go into the host cell. So at the same time you also edit the wrong properties of the viral DNA, because you don't want other diseases transferring, because we also use the HIV virus also for the as a vector. So you edit that, you insert into the host cells DNA. So by either you can do intracameral injections, you can do intravitreal injection if you are targeting retinal cells, retinal ganglion cells. So it gets incorporated into the messenger RNA, which goes and replicates into the DNA and sells the faulty DNA. This is how the gene transfer therapy works. Now editing means it does the partial editing, only focuses on the portion which is wrong or faulty. Now, what are the delivery system and target issues available in research at this point in time? We have 6 delivery system, namely adenoviral associated, LV, that means it takes care of the feline immunodeficiency virus and human immunodeficiency virus, HSV. Then we have a new liposomes, that is nanoparticle, and full DNA. These are the 6 delivery system available. We have 2 one is anterior delivery system, that is we are targeting the trabecular meshworks, and we have posterior delivery system, that means it targets the retinal ganglion cells by intravitreal injections. So here in anterior segment, basically where it works on trabecular meshwork, all the research animal models have found that LV, that is working on feline immunodeficiency virus or HIV virus, have a better chance, and one of the studies have shown that and I will quote some of the Kaufman has done lots of research on the gene therapy, and I'll give a couple of examples. And where the posterior segment delivery system is concerned, the intravenous delivery, usually it works by the adenoviral, that works much better, and some of the studies have shown that one intravitreal injection in the rat, it results up to the 72% of the transaction efficiency, and it works by the expressing up to the 3 to 4 months. Now, how it translates into clinical practice is long way to go, but the research is going on. So this is one of the example where they had used the feline virus for the trabecular meshwork to do increase the IOP reduction ability. So this is a Kaufman, as I mentioned, feline. What they had done was to use the lentivirus gene cassette for the prostaglandin biosynthesis, which increases the IOP reduction ability, and one injection had effect for the 5 months. They also had done, what you can do is that because your host DNA has a enzyme which try to degrade these molecules. So what we can do when you inject the gene, we also can disable these proteomeres which can degrade the particles. So they had done the both way, and this is with the proteasome blocking agents, and you can see that the collateral channels works much better and for longer time. The other models which works on the IOP regulation of trabecular meshwork using the urokinase gene expression, there are lots and lots of data. I have just given 2 examples because in 10 minutes I cannot cover everything. So to conclude, the glaucoma is complex gene mechanism, so is difficult to identify by one gene, one therapy is not possible. The second, screening is limited at this point in time. It should be restricted only for a very selective group of certain juvenile open angle glaucoma and congenital glaucoma. The mass screening is definitely not going to be useful, just like mass screening what we are talking about the diagnostic test. And third, the gene therapy has lots and lots of potential. It would be for both IOP, reduction, regulation by trabecular meshwork, and ganglion cell protection by intravitreal injections. Thank you.
Thank you, Dr. Rajul.